In 1988, the Morris worm crippled the early internet by accident, infecting 10% of all connected machines within 24 hours. Today, we face a theoretical horizon where that same logic of self-replicating malicious code jumps from silicon to carbon, treating human cells as the hardware and our DNA as the operating system. If a computer virus could reprogram your genetic code, the definition of an antivirus software would shift from a digital firewall to a literal immune system overhaul. This is not science fiction, but an emerging field of security called cyber-biosecurity, where the vulnerability isn't just your laptop, but your very biology. The fundamental language of life is digital. DNA is a 4-letter code composed of adenine, cytosine, guanine, and thymine, functioning as a set of instructions for the assembly of proteins. Because this code is readable and writable, scientists now use DNA synthesizers to print genetic sequences much like a desktop printer produces a document. Researchers at the University of Washington demonstrated that a computer file could be encoded into a physical strand of DNA. When a gene sequencing machine analyzed that DNA to turn it back into digital data, the resulting code contained a malware exploit that took control of the computer processing the sequence. This was the first time a biological molecule effectively hacked a digital computer. The terrifying inverse of this experiment is the biodigital bridge, where a computer virus escapes the screen to rewrite the user. To understand how a computer virus could reprogram a human, we have to look at how modern gene therapy already functions. We currently use modified viruses as delivery vehicles, essentially stripping out the pathogenic parts of a virus and replacing them with beneficial genetic instructions. This is how many new treatments for cystic fibrosis or certain types of blindness work. A malicious actor wouldn't need to invent a new biology. They would simply need to hijack the existing infrastructure of synthetic biology. In a world of decentralized biomanufacturing, a hacker could penetrate the servers of a pharmaceutical firm and subtly alter the digital blueprints for a vaccine or a therapeutic protein. Instead of the intended cure, the facility would unknowingly mass-produce a genetic payload designed to suppress a specific immune response or trigger the overproduction of a harmful hormone. This is a supply chain attack, where the end user becomes the compromised hardware. The potential scale of such an attack is unprecedented because biological systems are self-replicating. A digital virus on a network requires a connection to spread. But a biodigital plague leverages the natural mechanics of infection to distribute its code. If a hacker created a synthetic pathogen that hides within the human genome in a dormant state, triggered only by a specific environmental cue or even a radio frequency, we would see a delayed action malware for the body. This mirrors the behavior of a Trojan horse or a logic bomb in computing. A sequence could be designed to remain silent until it detects a specific chemical signature in the bloodstream, such as a common antibiotic, at which point it begins producing a neurotoxin. This creates a terrifying scenario where the medicine intended to help becomes the key that unlocks a lethal biological process. The barrier to entry for this kind of biohacking is dropping rapidly. 20 years ago, sequencing a human genome cost $3 billion and took a decade. Today, it can be done for a fraction of that cost, and in just a few days, it costs less than $1,000 and can be done in a single afternoon. The rise of DIY biology and community labs means that the tools for genetic manipulation are becoming as accessible as the tools for writing software. While this democratization of science promises incredible breakthroughs, it also eliminates the traditional gatekeepers of high-risk research. A lone actor with a high-end laptop and a mail-order DNA kit could theoretically design a genetic sequence that targets specific phenotypes. This introduces the concept of ethnic or individual-specific bioweapons. If a database of leaked genomic data from a site like 23andMe were cross-referenced with malicious code, an attacker could design a pathogen that only activates when it detects the unique genetic markers of a specific family or population group. It is the ultimate targeted exploit. A biological sniper rifle instead of a digital shotgun. Beyond physical harm, the biodigital plague could target the human brain's neurochemistry. Our moods, memories, dots, and cognitive functions are the result of complex chemical interactions governed by genetic expression. If a virus could alter the expression of genes responsible for dopamine or serotonin regulation, it could effectively remote control the mental state of a population. This isn't just about making people sick. It's about altering human behavior at scale. Imagine a strain of code that induces extreme lethargy or heightened aggression. In a digital to biological attack, the goal might not be death, but the total destabilization of a society's mental health. The intersection of CRISPR technology and digital connectivity makes this even more volatile. CRISPR-Cas9 is a tool that allows for precise editing of DNA by cutting out specific segments and replacing them. If automated CRISPR systems are connected to the cloud to receive updates or diagnostic data, which is increasingly common in professional research environments, those systems become vulnerable to remote hacking. A vulnerability in the software of a bioprinter could lead to the unintended insertion of a backdoor into the human genome of anyone receiving a treatment from that machine. This creates a permanent inheritable vulnerability, unlike a computer you can wipe or a password you can change. A genetic edit integrated into your germline would be passed down to your children. We are talking about the possibility of a multi-generational malware infection. The defense against such a threat requires a complete reimagining of national security. Traditional borders and physical lockdowns are ineffective against a threat that travels as a digital file and manifests as a biological agent. We would need a global network of biosensors, essentially a firewall for the physical world, capable of detecting synthetic genetic sequences in the air, water, or soil in real time. This would necessitate a massive leap in data processing, likely requiring AI to monitor trillions of biological data points to spot the signatures of engineered code versus natural evolution. There is also the ethical minefield of genetic privacy. If our DNA becomes a surface for cyber attacks, the government would likely demand access to our genetic data to provide protection. This could lead to a future where citizens undergo mandatory genetic scanning, much like we currently go through metal detectors. The risk is that the cure for the biodigital plague becomes its own form of surveillance state. We must also consider the accidental biodigital plague. As we rely more on AI to design new proteins and drugs, we face the black box problem. An AI might suggest a genetic sequence that solves a specific problem, like carbon sequestration or plastic degradation, but contains unintended side effects that mimic a viral outbreak. If that AI-generated sequence is released into the environment without proper sandboxing, the digital equivalent of a high-security lab, it could propagate through a global ecosystem before we even realize it was a mistake. This highlights the need for air-gapping biological production. Keeping the digital design phase entirely separate from the physical synthesis phase is the only way to ensure that a server breach doesn't result in a biological catastrophe. In an interconnected world, a true air gap is nearly impossible to maintain. Even the most secure facilities usually have some bridge to the outside world, whether through a maintenance laptop or a compromised USB drive. The history of cyber warfare, from Stuxnet to the recent ransomware attacks on hospitals, shows that no system is truly impenetrable. In the context of biology, an unpatched vulnerability isn't a crashed server. It's a global pandemic. Our legacy systems, our own ancient, evolved genetic blueprints, never evolved to handle the rapid, targeted modifications that digital tools enable. We are running billion-year-old software on hardware that is suddenly being exposed to the hyperspeed of digital evolution. To move forward, we must treat biological data with the same intensity we treat nuclear secrets. The convergence of IT and BT, information technology and biotechnology, is the most significant transition in human history since the Industrial Revolution. It holds the potential to end all disease, but it also provides a roadmap for the first truly programmable weapon. The ultimate question is whether we can build a digital immune system fast enough to stay ahead of the code that wants to rewrite us. We are no longer just users of technology. We are increasingly the medium in which that technology lives. Protecting that medium is the defining challenge of the 21st century. If you recognize the gravity of this shift, consider how we might regulate the production of synthetic DNA without stifling medical progress. The code is already being written, and it's up to us to decide what it says.